7-day read an article challenge. This is a challenge that is built for developing mental strength, and in this case I'm taking a challenge of reading 7 days a scientific article each day. By the end of the week, I hope to have more information about different subjects, increase my memory function, increase the processing of information, and making reading of scientific articles fun for me. Today is the day 3. The scientific paper that I'm going to present today is about the anti-transporter known as Kia3, which is a potassium channel anti-transporter, and we are going to talk about regulation that can be mediated on this protein based on a mutation. So here I have the name of the paper, the name of the authors, and their affiliations for you to take a look into the paper if you are interested. The word Kia stands for potassium, that is symbolized by K plus, exchange, antiporter. So K is from the symbol of potassium, E from exchange, and A from antiporter. There are 3 different kinds of the Kia proteins present. Kia1 and Kia2 are present in the chloroplast envelope, whereas Kia3 is present in the thylakoid membrane of the chloroplast. Now, today's topic revolves surrounding the Kia3. So let's get into the introduction. Generally during photosynthesis, the energy equivalents that are produced are NADPH and ATP during the light reaction of photosynthesis. This accumulation of NADPH and ATP are the major sources that finally lead to carbon dioxide assimilation. In order for the NADP to get reduced to NADPH, there needs to be transfer of hydrogen ions from the stroma to the lumen of the thylakoid membrane via the processes of water splitting and transfer of electrons by the Q cycle in the cytochrome B6F complex, which actually results in the development of a proton motive force across the membrane based on the accumulation of the ions across the membrane. Proton motive forces are composed of 2 components. One is the electric potential component and the other one is the pH. Now, depending upon the concentrations of ions present across the membrane, the delta pH can vary. This amount of delta pH that is present is important because it leads to the ATP formation that will happen eventually. However, the ratio of ATP to NADPH that is produced via just the linear electron flow does not account for the ratio that has been observed for ATP to NADPH produced, which means that there should be some other alternative methods for the electron flow happening. pH does not only have roles in ATP synthesis, it is also involved in other processes. Delta pH can affect the PMF, remember? So, in plants, it has been seen that the delta pH can also affect the photoprotective mechanism in presence of high intensity of light. Delta pH can induce the QE component of NPQ of chlorophyll fluorescence in the photosystem too, and this is actually triggered by luminal acidification. If the pH of lumen is low, the activity of cytochrome B6F is downregulated, slowing down the rate of electron transport that happens between PS2 to PS1. And this is an important method for regulating the photosynthesis in fluctuating light. The reason why the pH can vary is because of ion fluxes across the thylakoid membrane, and thus the ion fluxes can regulate the PMF across the membrane. In presence of light, there could be a flux of hydrogen ion and uptake of potassium ion by help of antiporters such as Kia. Kia3 loads potassium ions into the lumen by using delta pH formed across the thylakoid membrane during illumination. The reason why we are discussing about this paper is because in this paper the authors have isolated and characterized a mutant allele of the antiporter. This mutant allele we will be calling the disturbed proton gradient regulation allele, or DPGR, in which the regulation of Kia3 activity was modified, and this was compared to a knockout allele of Kia3. The knockout allele will be considered to be Kia3-1 for future references. By comparing them, it was seen that how the regulation of PMF can be contributed by Kia3 during induction and in different light intensities. In order to study the regulation of the PMF formation via alternative electron transport not depending upon LEF or linear electron transport, mutants with high chlorophyll fluorescence was screened, which would indicate low NPQ. Conditions were chosen in order to enhance activity of alternative electron flow pathways. Carbon dioxide removal prevents the linear electron flow or LEF, and reduced oxygen blocks photorespiration. For this experiment, second generation mutant seedlings were chosen and illuminated by actinic light at 50 micro-Einsteins in an environment that consisted of 5% oxygen and air that was free of carbon dioxide. 45,000 seedlings were screened, and from there 3 mutant kinds were confirmed on basis of high chlorophyll fluorescence. There was one allele responsible for this kind of effect, which was due to the protein gradient regulator kind 5, also referred to as PGR5, and the other one will be referred to as, the allele name stands for chlororespiration protection 2, CRR2. This kind of screening is suitable for identifying mutants that are involved in alternative electron transport pathway. Another mutant with respect to PGR5 and CRR that was identified is the mutant called DPGR mutant, about which this whole paper is, and this is defective in PS1 cyclic electron transport pathway. And today we are going to talk about mainly this mutant. When the DPGR5 mutant seedlings were experimented with under the exposed actinic light, it was seen that they have a high level of chlorophyll fluorescence that was being expected, as you can see in the figure. The high amount of fluorescence can be lowered by an alternative electron sink in the artificial air or by induction of QE. The biggest question is that how is QE induction of NPQ affected in this DPGR mutants? For doing that experiment, the same kind of environment that was used for the previous experiment was used, along with another ambient environment of air. The first kind of environment that was used was with 2% oxygen, and in that 2% oxygen levels, it was seen that the NPQ was decreased in case of the DPGR mutant, which is shown by the red bars in the graph of the graph A, compared to the wild type, which is represented by the black graph. Whereas in the ambient air environment of 21% oxygen, we see that the DPGR level was initially low at the first minute, but however, it came back to the level of the wild type after 4 minutes of light exposure, as you can see in the graph B. Now, the mutant PGR5 is defective in antimycin A sensitive pathway of the photosystem 1 cyclic electron transport, and the mutant CRR2, which is also referred to as CRR2-1, it's defective in the antimycin A resistant pathway of the photosystem 1 cyclic electron transport. These two mutants were compared along with the double mutants that consisted of these mutants along with DPGR. In case of single mutant of CRR2, it was shown that there was low NPQ induction in 2% oxygen as compared to the DPGR mutant, but this decrease was not seen in the 21% air environment. NPQ was decreased further in case of the double mutant of DPGR along with CRR as expected in both of the environments. Remember here that CRR is the kind of mutant that actually is defective in the antimycin A resistant pathway. So this kind of explains the behavior that we see in case of the CRR mutant and the CRR DPGR mutant. In case of PGR5 single mutant, which is an antimycin A sensitive mutant, in that case, it was seen that the size of NPQ was further reduced even in 21% oxygen, unlike that we had seen for CRR. And in DPGR PGR5 double mutant, the size of NPQ was reduced more than the single mutant for PGR5, which was also seen at 2% oxygen level. So these results indicate that the induction of NPQ is disturbed in DPGR mutant in artificial air, and this disturbance is most likely via mechanism that is independent of the cyclic electron transport, because we check the cyclic electron transport by mutating the other two kind of protein transporters that are present, PGR5 and CRR2-1. The reason why in the low oxygen environment 2% oxygen was used rather than 5% oxygen was in order to characterize further the DPGR characteristics based on chlorophyll fluorescence, in which the chromosome 4 was substituted by that of LER. The analysis of the resulting F2 plants showed that the DPGR locus was present within 2910 kb region, which included also the centromere of chromosome 4, as you can see in the figure of the slide that is present on the top. The DPGR sequence was compared to that of the wild type sequence, and there was a lot of nucleotides that were different in between the two. One such difference was also seen in the nucleotide region that encoded for the KIA3 protein, in which due to the change of a specific nucleotide base, the protein that was encoded by this part had an amino acid change from glycine to arginine, and this change could cause the disturbed NPQ regulation. This kind of mutation that is seen that causes the disturbance for DPGR would be known as a missense mutation. The people or the authors also did experiments where they inserted tDNA mutant alleles of the KIA3 knockouts and analyzed the effects with respect to that of DPGR. KIA3 consists of two domains. The protein has a cation proton exchanger, also known as CPA2 domain, and a putative potassium nucleotide binding domain known as KTN. DPGR causes mutation in the transmembrane region of CPA2. There are nearly 11 transmembrane regions present in the CPA2, and only one of the transmembrane region is affected. Protein blot analysis of KIA3 was done for the wild type, the DPGR mutant, and the KIA3 knocked out mutant. Of course, for the knocked out mutant KIA3, there was no presence of KIA3 that was observed for the fraction. However, for the mutant DPGR and the wild type, we see that there is presence of KIA3 of almost the same level. The upper band for the protein that is observed in the immunoblot is for the whole protein of approximately 80 kilodalton, and the lower band that is present is a product for alternative splicing of the protein. You can also see the bands in the Kumasi blue stain for protein analysis in the bottom figure of this slide. In previous research articles, there was a suggestion that the KIA3 protein helps in the import of potassium plus to the lumen and efflux of hydrogen ions from lumen to the stroma. However, in the DPGR mutant that we just saw, there was a low NPQ, and that brought in the question that if the topology that was thought to be present for KIA3 is the correct topology or not. Based on the Aramaenon database, KIA3 is thought to harbor 12 to 13 transmembrane domains. However, as we had discussed earlier, the KIA3 protein has two main domains, one facing the stromal side, known as KTN, and the other one the CPA2. Now, the KTN domain can be dissolved by using a protease such as trypsin. So, in order to know the location of KTN and how the topology of KIA3 is present in the thylakoid membrane, the intact thylakoids that were isolated from the plant leaves from the wild-type plant were digested using the trypsin. If trypsin digests the KTN domain completely, in that case, the KIA3 antibody that was raised against the KTN domain will fail to detect the KTN and also fail to detect the KIA3 protein. So when the protein, for which you can see the initial molecular weight band as in figure A, was treated with trypsin, within 10 minutes the band for KIA3 disappeared. However, the lower band, that is due to the alternative spliced product of the KIA3 gene, we can still see it present. However, with increasing time, it starts disappearing. If we compare these bands with respect to the marker used for this experiment, which was PSBO, which is a marker for the thylakoid lumen, we can see that the KIA3 was not detected due to the digestion of the KTN domain that was exposed on the stromal side. So now we definitely know that the orientation of KIA3 or the topology of the KIA3 in the thylakoid membrane is such that the KTN is present in the stromal exposed side, and there is a influx of potassium into the lumen and an influx of hydrogen ions to the stroma that is happening as denoted in the figure B. To remind you briefly, the KIA3-1 allele is a knocked out allele of KIA3, which means it gives rise to the plants that would not have the KIA3 antiporter. Whereas the DPGR allele is the allele that would cause phenotypes to have the KIA3 protein but not function as much as the KIA3 would do. So the NPQ for the wild type protein, the DPGR and the KIA3 knocked out were analyzed, and the results suggested that the NPQ phenotype for the KIA3 mutant was completely different from that observed for the DPGR. In fact, it had the opposite phenotype. When compared, we see that KIA3-1 mutant has a reduced NPQ as compared to the wild type. However, the DPGR mutant has lesser NPQ than that with respect to wild type or the KIA3 knocked out mutant. The NPQ phenotype in DPGR that was seen was accompanied by the reduction in the photosystem II yield, which was not seen in case of the KIA3 mutant. And the results for that has been reported in the supplementary paper, and I'm not going to go through it, but I would suggest that you should take a look into the data from there. For NPQ characterizations, the experiments that were done using overnight dark adaptations showed in wild-type plants the NPQ was transiently induced to a maximum level within the first minute, followed by rapid relaxation in the next few minutes. This phenomenon is not present in case of DPGR, that is shown in the graphs using the crimson colored band. The wild type in the graphs are mentioned in the black band that you can see. And you can see the difference in the induction at the very first time point in the graphs. Just to remind you, these results were from the artificial air environment that was produced, given for this experiment, where there was no CO2 and the percentage of oxygen was very low, up to 2%. In the KIA 3-1 allele mutant, which was the knockout mutant, it was seen that the transient NPQ induction was unaffected. If you look at the spectra, it's pretty much similar to that of the wild-type spectra of NPQ, suggesting that the KIA3 is not required for this process. This result suggests that the regulation of KIA3 is disturbed somehow in the DPGR mutant. When higher light intensity was provided of up to 110 micro-Einsteins, in wild-type plants, it was seen that there was a rapid induction to a maximum level within the first 2 minutes, and the relaxation was much more gradual as compared to the lower intensity light of 50 micro-Einsteins that were provided initially, which actually reflect that this gradual relaxation was due to the full activation of the Kevin-Benson cycle, and this is absent in the DPGR mutant. However, a comparable level of NPQ was induced at 10 minutes after the induction of photosynthesis in both the DPGR and the wild type. In contrast, a similar level of NPQ to wild type plants was induced within 2 minutes in the KIA3 knocked out phenotype. The relaxation of NPQ was slower in the KIA3 mutant plants than with respect to the wild type plants, which resulted in higher NPQ phenotype for the KIA3 mutant. The level of NPQ was similar at 10 minutes after the induction of photosynthesis amongst all the 3 phenotypes, genotypes of the plants present: the wild type, the DPGR, and the KIA3 knocked out mutant. As showed in the slower relaxation of NPQ in the dark phase. Phenotypes of the 2 mutant alleles heterozygous plants were studied in both the artificial, which is 2% oxygen air, and ambient, which is the 21% oxygen air, was studied to verify the dominance of the DPGR mutant. Heterozygotes of KIA3 mutant allele displayed levels and time course inductions of NPQ similar to that of the wild-type plants, as you can see from the bottom figures. In both artificial and the ambient air. But the DPGR heterozygotes that were present had reduced NPQ induction that was similar to what we have already seen for the homozygous DPGR. In case of ambient air, the levels of NPQ in DPGR heterozygous was intermediate between that in the wild type and the homozygous DPGR plants, which suggests that DPGR was semi-dominant in the ambient air condition. DPGR mutations showed a dominant nature in the ambient air, probably due to the disturbed regulation of the KIA3 activity, whereas in case of KIA3 knocked out mutant, it showed the phenotype of having a recessive nature. To complement this study, further experiment was done by putting in the full-length genomic sequence of the KIA3 gene from wild type into the DPGR mutant, and based on that combination, there were 6 independent T2 lines that were finally analyzed at the different air environments. In artificial air, NPQ recovered to different intermediate levels between those of the wild type and the DPGR plants in the lines that are shown in the graph below. Line 1 and 16 had high complementation, whereas in line 15 it was not present. To that extent, based on the induction of NPQ in ambient air, the different lines were classified into 3 groups. The first group, where the complementation was good, such as in 1 and 16, the NPQ was enhanced, as in the knocked out allele, where there was no protein present. Co-suppression of the KIA3 genes, both the transgenic wild type gene and the endogenous DPR genes in line 1 and 16 were probably producing a phenotype that was similar to the KIA3 knocked out allele in both the artificial and the ambient air environments. The second group consisted of the line 15 that showed DPGR-like phenotype in ambient air, and we can see based on the blot present in the bottom panel figure that the KIA3 protein level did not change dramatically. And then there was the third group consisting of lines 21, 10 and 11 that showed the wild type like phenotype in the ambient air. So depending upon the different crosses, we can see how the KIA3 expression of phenotype can vary among the different lines. The next thing to know was if the PMF of the 2 different kinds of alleles phenotype that we are looking at would change or their size would change during the induction of photosynthesis, causing the change in NPQ induction eventually. And that could be studied by using the electrochromic shift assays, But the result that was obtained from the ECS spectra suggested that there was similar pattern between the wild type and the two different mutant alleles of DPGR and the KIA3 knocked out in course of the PMF induction. Therefore, the phenotypes of these alleles observed in NPQ induction were not due to the size of the PMF, but because of how the PMF partitioning was done in between the two components of PMF, that is the pH change or the electropotential change. With this, I would bring to the conclusions that I obtained from the paper. So generally, KIA3 is a thylakoid membrane localized antiporter protein that helps in the transfer of potassium to the lumen and hydrogen to the stromal side by the help of this antiporter, and that happens due to the presence of the proton motive force that is present in the membrane. The mutant allele of KIA3, known as DPGR, which also stands for disturbed proton gradient regulation, reduces the NPQ in artificial air environment that has no carbon dioxide and low oxygen. This phenotype is enhanced in presence of other mutants that affect the photosystem 1 cyclic electron transport, such as PGR5 and CRR2. The DPGR mutation affects the 11th transmembrane domain of the KIA3 protein that is present. In presence of ambient air, there is a reduced NPQ that was observed during the induction of photosynthesis in the DPGR mutant. Whereas for the knocked out allele KIA3 one knocked out, there was a higher NPQ phenotype opposite of what that was observed for DPGR. Based on the experiment done in the ambient air, it was understood that the membrane topology of KIA3 indicates a proton efflux from the thylakoid lumen to the stroma, as also evident from the protease digestion experiment that was explained in this presentation. The DPGR heterozygotes that were produced showed a semi-dominant phenotype in case of artificial air that had 2% oxygen and a dominant phenotype as seen in the ambient air of 21% oxygen. In this case, we have to remember that if it was dominant in the artificial air, in that case it would suggest that there was a complete distortion of the KIA3 due to the DPGR mutation, but that does not occur over here in this case. In the DPGR mutant, the protein level of KIA3 was unaffected, but there was a downregulation of its activity, and that was probably because of the disturbance that was produced. Thus, we now know that DPGR mutation on the KIA3 could lead to the regulation of the KIA3 activity and change the NPQ level that was produced due to the KIA3 activity, and this is necessary for optimizing photosynthesis. That is why it is important to study how the mutation of DPGR has affected the KIA3 activity as shown in this paper. I enjoyed reading this paper on the third day of my 7-day challenge of reading a scientific article per day. If you are taking this challenge, let me know how was your experience over the days. Once I have finished the 7 days, I will let you know how it goes. Here is the chart on which I have been marking the days that I have read. You can download this chart and print it for yourself to have a note on which days you are reading it. If you like this video, please put a thumbs up. Thank you for listening to this presentation. And if you like or dislike this video, please put your comments down. Please let me know how I can improve these presentations. And subscribe to my channel to see more similar kind of activities that I do in my channel. Have a good day ahead. Bye till tomorrow.